Okay, let me get going. Thanks everybody for coming. This is our last Friday. Yes, one more. I'm coming. So I'm going to largely stick to the minimalism content. Brian DePasquale here today. Speaking, they started out a while with you, and I was really excited to connect with because one of the few people that understand how to train the current spiking networks. I think that as we think about finding people this all very just applied neuroscience, that's something that we're all going to have to face.
Thanks so much. It's really great to be here. I appreciate the invitation, and I'm glad to come talk to you about all the work that we're doing over at BU. I'm going to give a little bit of a an additional introduction, just because I feel like everyone always is like, I'm really happy to be here, and I actually am really, really happy to be here for a reason that I'd like to tell you. This is like my lineage. I came from Columbia, where I did my PhD, did my postdoc at Princeton, and then, as Bernardo said, I just started my lab at BU, which might give you the impression that I'm a New Yorker, but I'm actually not a New Yorker. So this is a map of Boston. Can we bring the lights down like a hair, maybe? This is where we are right here. And there's a small village in Newton, about 5 miles west of here, called Nonantum. And I'm going to press you right off the bat to see who's an actual real Bostonian here and who can name the nickname for this village.
Nickname for what?
For Nonantum.
I don't know the answer.
What's that? The lake. Yes, it's called the lake. So this is a very special place to me. This was a site of a lot of Italian-American immigration in the early 1900s. This is my grandfather, who didn't finish 8th grade, and he grew up in Nonantum, where my entire family grew up. So I'm actually much more closely associated with Boston than I am with New York. And so it really is a very, I'm really, truly glad to be here, because my grandfather is no longer here, but I think he would be kind of flabbergasted to know that his picture was being shown at an academic talk at Harvard, having never finished 8th grade. So with that, I'd like to tell you about the work that drives my research interests and the research interests of my growing group at BU. And the big question that I think about mostly is how can we build mechanistic models of neural computation? And so I'd like to unpack what I mean by that a little bit by walking through what I think is one of the great successes in computational neuroscience, and that is of the ocular motor integrator of the goldfish. So I think this is a very impressive set of studies, because it shows how we can go from experimental work to a reasonably sophisticated mechanistic model within roughly the span of a decade. So how do we begin? Well, of course, we have to start with observations of real neurons. So what I'm showing you here is the firing rate of a population of neurons. This is in the hindbrain of the goldfish. And the experimentalists recognized that these neurons' firing rate needs to code a population code for eye position. So 5 years later, Sebastian Sung came along and developed what I'll call, throughout this talk, this idea of an abstract model. So this is a model that will condense or kind of give us the insight into how this neural population works without actually building it into a biophysical or a real mechanistic model. And so the idea behind this model is that we can represent the activity of all these neurons in a state space, which I'm showing you here on this as a plane. And as the eyes move, the population of neurons will change their firing rate, and that will correspond to movement along a constrained line within this state space that will allow us to understand how the eye position can be encoded in as a one-dimensional variable in this neural population. And then as the eyes stay still, there's no more movement along this line. So 4 years later, Sebastian went on to build a biophysical neural circuit model of how this works. And the intuition here was that through recurrent excitation, you could generate a neural circuit that gave rise to persistent excitatory firing that would allow you to instantiate this abstract model into a mechanistic model. So in my mind, this really is a success story. Went from the beginning to the end within a little over a decade. And I think ideally, this is what at least I would like to do for as many neural systems as possible, try to grasp how we can build a real mechanistic model of the computation of interest. So what are the problems with that? Well, this is not going to be straightforward, it doesn't seem. So I'd like to suggest that there's essentially 3 challenges that we need to overcome to do this. And this is roughly the 3 different kinds of things that I work on in my group. The first challenge is that neural responses are quite a bit more complex than the ocular motor integrator. So we're going to need to develop methods to understand how this complexity relates to the complexity of behavior. The second is that neural responses tend to not only be related to external covariates, but a lot of internal variables that we often don't have access to. So we'll have to develop methods that can deal with this challenge. And the third is that it can be often not entirely clear how to map these abstract variables onto biophysical neural circuit models like Sebastian did in the ocular motor integrator. So my lab really focuses on trying to bridge machine learning and mechanism through these 2 different avenues of investigation. In machine learning, we tend to focus on building models that we can deal with neural complexity and understand how neural complexity relates to internal variables represented in the brain. And in my mechanistic modeling work, we focus on how we can map these abstract models that we're learning with machine learning into mechanistic models of neural circuits. So I'll tell you mostly about the mechanistic modeling today, about how we construct recurrent neural networks of spiking neurons that can essentially perform most types of computations that we're interested in non-spiking systems. And I'll tell you a little bit about some of the machine learning work we're doing that I did as a postdoc, and now we're continuing on in my lab at BU. So this is the title of my talk, which was Constructing Spiky Networks as a Foundation for the Theory of Manifolds as Computational Substrate. And really, there is kind of 2 parts to this effort. One is how do we build these models? And 2, once we built them, what can we do with them? And how can it allow us to bolster the kinds of theoretical concepts that others in the community are formulating? So this is the picture that we mostly start with as neuroscientists. We have a population of neurons responding and some kind of behavior that is being generated based on that neural activity. So this is a recording from the motor cortex from Mark Churchland's lab. And what is being shown on the top is the velocity of this animal's arm as it's moving. And an emerging perspective on how these 2 things relate to each other is via this abstract model that we typically refer to as a neural state space model. It goes by, I think, of a lot of different names depending on which way you've kind of come into the field. Latent variable models and statistics. A lot of people like to think about it in geometrical terms like neural manifolds. I'll typically refer to as neural state or using Mark's preferred term of factor. But the basic idea is that we have some kind of lower dimensional neural state that's described in a space of latent factors, or you might call it a manifold. And that the key computations that allow a neural system to perform the behavior that it needs to are represented by activity patterns that are constrained to live on this manifold. And this is the right way to understand the mechanism by which the neural network is performing the job that it is performing. And so what I'd like to tell you about today is neural circuit models that we were developing of neurons that spike, that instantiate these ideas into the model itself. So it's perhaps not necessary to belabor this here, but this is probably one of the most widely used abstractions in systems neuroscience right now across a variety of different systems. And it seems as though people have really latched onto this as an effective way to understand how neural circuits work. But this has, I'd say, left a little bit of kind of hesitation by some in the community as to wondering if this is a real abstraction that is actually going on in a neural circuit, or is this just like kind of pretty pictures that we're drawing that help us understand how neural systems work, or is this how this system actually does work? And the challenge, of course, is that you can't typically you can't measure these directly. You have to infer them through a model inference procedure, and usually in a situation where you have incomplete access to the entire system. And so what we really sought to do is build a neural circuit model that could describe exactly what these signals are in a biophysical neural circuit in order to give them a face, so that any remaining hesitation by any of the deepest naysayers could be kind of pushed away. And as I said, one of the big challenges is that these concepts are principally defined, I'd say, in a statistical sense. And what I mean by that is that we typically think of some low-dimensional set of functions, which we refer to as factors. And we think of them as sort of being reflected in a neuron, which gives rise to some kind of underlying firing rate of that neuron. And we typically model the spikes that we see based on that that neuron's underlying firing rate as being probabilistically generated as a sort of through some kind of random process. And this means that we're essentially constructing a statistical model of what we're seeing. We're doing inference to learn these factors from essentially what we're calling a random observation. We're not constructing them as being mechanistically generated within the neural circuit itself. So what we really sought to do is kind of flip this the other way and say, can we build a model where we have a deterministic neural system that is constructing these latent variables within its synaptic connections? And we know, because we've built it, that this is exactly how this system computes. And so a few of the key points that I'll walk you through in this part of the talk is that we introduced a flexible method for constructing functional spiking neural network models. And this flexibility is important because it not only allows us to use data and explicitly represent what factors are in spiking models, but it essentially allows us to constructing spiking models that can do virtually anything that you could do with a non-spiking system. And so, I mean, I feel like a perennial complaint that I would get in neuroscience papers would be, well, that's really cool, but can you do this with a spiking model? And the answer is just yes now. We don't really have to quibble with reviewers anymore. The answer is yes. Just Brian, we figured it out. Just maybe more difficult. You maybe need more parameters. Maybe it's going to take a little bit more time, but I think we basically got it. And we build these models so that the idea of the latent factor is patently mechanistic within the synaptic connectivity of the circuit itself. And really, the hope there is to give these models, these concepts that some have sort of wrung their hands about, a real face in a biophysical neural circuit. And we do this in spite of various other really important aspects of neural activity that we've observed empirically, principally that we often see that neural tuning is heterogeneous, and we see a great deal of spiking variability in neural responses. If we cannot construct a model that generates these latent factors in the face of these, then we're not quite doing our job. And then once we built these models, we can use this to really give these terms that we've derived statistically a mechanistic face. And two of those, as I said, one principally is the latent factor. And what we kind of get along for free is another term that maybe some have not recognized as actually being a statistically inferred term, which is the firing rate, which has been another kind of hairy thing for theoretical neuroscientists to give a face to. And what we can show is that once we build these models, the firing rate really just emerges quite naturally. I'm very happy to take questions at any point. I think it's a much more fun and enjoyable time to interact with the audience. Are there any questions so far about what I've said? I mean, it's all pretty straightforward. So, but I'm happy to take any in reflection. Okay. So how will we build this thing? Well, we're going to start with a simplified model of a single neuron. This is a leaky integrate and fire neuron, which is essentially the simplest model neuron that we have that generates spikes. We're going to integrate, we're going to sum the synaptic input from the other neurons in the network that I'm not showing you here. This neuron should be familiar to most of you, but it's going to integrate its input. And when that integrated input crosses some threshold, that neuron will spike. And what's really important to appreciate here is that there are no factors in this model yet. This neuron does not have a firing rate, and it is not going to produce spikes in a probabilistic manner. These spikes are going to be determined by a deterministic dynamical rule. And that means that any properties that is endowed with that look like these kinds of things, like probabilistic spiking, are not going to be due to it's going to reflect what looks like probabilistic spiking, but it's actually not going to be due to a stochastic process. So we will take these population of leaky integrate and fire neurons. We're going to filter their spike trains through a synaptic time constant to generate this. Is there a pointer? I mean, I have one on my arm.
No.
No. That's okay. So that we can filter those spikes, and we'll get a filtered synaptic spike train, which we call S of t here, that we're going to sum through a set of what we call kind of pseudo synapses to construct the representation of the factors within the model. So this will be a linear sum, a weighted linear average of the spike trains to construct this low-dimensional representation of a target, of a signal. And then in order to generate a model that does this self-consistently, we're going to need to feed back those generated factors back into the model through another set of sort of what we call pseudosynapses U that will feed those factors back into the neural spiking population. So you might be noticing that this is beginning to look like a full recurrent neural network. We'll get to that in a second, but we've sort of contorted the recurrent neural network to sort of draw out where the factors live within it to kind of identify them more explicitly. But this mathematically is just a recurrent neural network. So this is the dynamical equation for this model. The key point here is that we're going to learn the set of pseudosynapses W with the recursive least squares learning rule. And we're going to train those synapses W so that the factors that the model produces, Y, match some target function Y, which will generate in a variety of different ways, depending on what we're interested in doing. So this is not a biologically plausible learning rule. It's just a way to construct a model that's going to do something that we want it to do. Hi, yeah. Hi.
For the general time network, you would expect that there are these types of one.
Well, so Y is not really a neuron here. It's like a linear neuron. So if I removed Y and then there was no if I removed Y and had each neuron connect to each other neuron directly, that it would be filtered by the synaptic time constant, which I'm kind of doing before the Y through that filter S. So I'm not showing the dynamics equations for S, but that's how you map the spikes into the synaptic filter. Is that clear? Yes. So you will see, I mean, you can see right here, actually, and you might start to notice what's going on here. If I just took Y and plugged it in there, I'm going to get U times W times S. And so the filter is kind of drawn out into the spikes on the S side, and then you're going to get this U times W. And you're probably already beginning to notice what this U times W term is looking like.
But the learning set is not good.
No, this is not a biological learning rule. No, not at all. I mean, I think it's very interesting how we could start to migrate into that space, but we sort of started from square one. Can we do this at all? Okay, so as I said, one of the motivations here is we want to construct these models so the latent factors are patently mechanistic. We've inserted them into the synaptic connectivity matrix to sort of give them a face. And now I'm going to illustrate that this actually works. So we're going to use some data from Mark Churchland's lab that we gathered from monkeys who are performing a cycling task. So this is a task that I think probably many of you are familiar with from the Churchland lab, but if you're not, this is a monkey who's basically moving a bicycle pedal with its arm to move through a virtual world. And my colleagues in the Churchland lab reported from a number of neurons in the primary motor cortex of monkeys while they're performing this task. So this is what these neural responses look like. This is the trial average firing rate of 5 neurons as the monkey is pedaling through this virtual world. And the Churchland lab has spent quite a lot of time analyzing these types of firing rates, doing dimensionality reduction on these data, and showing quite convincingly that by performing dimensionality reduction on these data, you can generate a small number of temporal functions called factors, which is completely sufficient for decoding much of the EMG activity of these animals. So, which is to say, doing dimensionality reduction on the rates is sufficient to construct the behavior. That's what we're going to do. We're going to take these firing rates, we're going to apply PCA to them and generate a set of 12 factors. And the implicit idea here is that those 12 factors is where the business end of computation is happening, and we want to now put that into a spiking model and get it to do the exact same task that the monkey is doing. So here's a train. Yeah.
Wait, can I just ask you, I know this is an older paper, I should know this, but I would have thought that the output dimensionality is quite low here, right? Am I wrong? I mean, how many dimensions are there in the motor activity or the EMG?
I mean, I think the dimensionality of the EMG activity for cycling, I think, is roughly 4. I mean, I mean, not surprisingly, it's pretty.
Almost higher than the output.
Yes. Yeah. So I think 95% of the variance was in the firing rate activity of the neurons was captured by 12 PCs, and my memory is that is more than enough to decode the EMG activity almost perfectly, which is perhaps a little bit surprising. So this is a trained model. What I'm showing you here on the right is not real neural activity. This is from a simulation that was trained to do this task. This is 100 neurons over 200 milliseconds. And what I'm showing on the right is the factors in the dotted line are the factors that were given to me from the data. And the blue line is the fit of the model to those factors. And you can see, unsurprisingly, the model learning procedure is working. It's able to construct those factors perfectly. And the key is that there's nothing, I mean, you can see there's clearly a quasi-periodic signal going on in the neural population, but the neural population is embedding those factors within its neural activity. The spikes reflect those factors. The factors are constructed from the spikes in this recurrent manner, and the network has successfully embedded this low-dimensional, 12-dimensional manifold into its spiking activity. And this is perfectly sufficient for decoding the EMG activity, just as it was true for the actual data. And so one thing I didn't...
Low-dimensional spatial.
Yes. Talking about the actual temporal signals themselves or the projections?
Is there in the simulated network, is there an external temporal signal?
In the
In the simulated network.
there is no external. This is an autonomous dynamical system.
Dimension.
Yeah.
Yes. And frequency then what's it then?
So the only temporal filtering that is being applied in the network, there's a temporal filter on the voltage, and there's a temporal filter on the synapse. That's S of T.
They are fixed, they're not trained.
Those are not modified.
With all this is autonomous oscillation.
Yeah, that's right. In principle, yeah, you could probably modify those through a.
So surprising that we generate the same frequency. Why?
Well, yeah, I mean, there's a little bit of, so the synaptic filter is about 100 milliseconds, which I think is reasonably well matched to the temporal dynamics of the signal. Well, I mean, the RLS is pretty powerful. And, you know, I'm essentially constructing a 12-dimensional signal from.
No, dimensional is in space, but the spatial structure you encode by training, so that's fine.
Yeah, I mean, it's, you know, a periodic oscillator. It's not terribly complicated.
So next question, should it fit the same frequency?
That's the target. I mean,
it's
target is that.
No, but they both match in this additional.
yeah.
You're using a fixed time interval, right, for your, like, your periods,
Yeah, you see,
rather than.
they're on top of each other.
So maybe like, so these are three factors from the model, right? So this is 3 of the 12 dimensions, spatial dimensions.
They're spatial, but you were showing temporal. What fits? Why
So this is the temporal dynamics of one of them.
Right.
And you're saying,
why the frequency match?
well, I mean, that if the frequency didn't match, then the learning would fail. I wouldn't.
It doesn't. Are you training also the frequency?
No, it's, I mean, at every moment, it's just saying, modify the synaptic connections to minimize the mean squared error between the target.
The entire.
I mean, it's running, it's an RLS, you know, RLS, so it's running in, yeah. I mean, you know, if you did regular old least squares between 2 functions, right? If you could construct it. And also you have to, I mean, one thing that's, you know, also perhaps relevant is the, you know, the spikes are being driven by the function that it just generated. So the factors are constructing themselves through the recurrent connectivity. So, and if the spikes are presumably oscillating at the same frequency that they're generating the factors for.
Right? I thought that the paper was a current network, which then you would pose a low rank.
Yeah, we're going to get there. Yeah, I did it backwards this way. Yes. So the way that we introduce spiking variability and heterogeneity is by including a second connectivity matrix into this model that so there's actually, I didn't tell you the full picture here. There's actually 2 connectivity matrices. And so I think the clearest way to illustrate this is to kind of unwrap the model in time. So what I'm showing you here on the right is the neurons at time t passing through these factors, and then the neurons at the next time step on the right. And it should be a little bit more explicit now that when you pass through these factors and you have these two pseudo matrices here, these pseudo connections, when you take the product of the two of them, this is essentially going to form what we call a low rank matrix, which is at this point becoming a very commonly observed feature in theoretical neuroscience, where you factorize a matrix into these this skinny, tall, and short, fat matrix. But what I didn't show you is there's actually a second matrix in here that I initialized the system with, which is a purely random connectivity matrix, which I called J0. And this is inspired by lots of work, and most initially Heim's work, showing that if you take a randomly connected neural system, it will generate close to Poisson-like spiking variability without really doing very much. So if you take it, wire it up randomly, you'll get a kind of spiking variability that we commonly observe empirically. So what's actually going on here is it's actually two matrices in this model. There's one that's random and fixed, and that's generating this intrinsic spiking variability that we'd like to see. And then we add on top of this low rank matrix that's learned, that's constructing the types of low rank, these low dimensional dynamics that we want the system to perform. So we can get a picture now of this variability by taking a slightly different look at our model. So what I'm showing you here is the activity of one neuron across many different trials of this system performing this task. And when we first started building this, Larry used to go around to all the experimentalists and show them a real neuron and show them this neuron. And he'd say, tell me which one's the real neuron. And I think that if I showed you this and told you this is a real neuron, a lot of you might think it actually is a real neuron. And so we really were very proud of this because we were able to simultaneously construct a model that was extremely stable in a set of a very confined set of directions, and then incredibly variable in a different set of directions that gave rise to this spiking variability. So you can quantify this across the population using a Fano factor metric. This was not fine-tuned to get the exact same Fano factor metric that is exhibited in data, but this is roughly the same. And we can take a slightly different view of this to try to understand how the activity in this random, how the activity due to this random matrix and this low rank matrix interact with each other. So what I'm showing you here on the right is the activity of the first two principal components, the first two factors, across many iterations of this model. And what I'm showing you in gray is those different individual trajectories. In blue is the average trajectory across all of them. And so despite the fact that there's quite a lot of spiking variability in this model, the spiking variability does not manifest as strong variability in this restricted subspace where the factors live. So it does induce some amount of variability in that space. You can see there's tiny little deviations in the gray, but it's mostly contained to exist outside of that restricted space. So with this now, we've sort of given, we've constructed a model that's going to give a nice face to what these latent factors that we've been building. And now this allows us to understand one other property of a neuron that has typically been statistically defined, which is the firing rate. So we can take a look at how these two different synaptic contributions give rise, what they look like into an individual neuron and how what their effect is on that neuron's output. So what I want to show you here is the one factor input for one neuron across two different trials. And what you should see is that the blue traces basically overlap with each other, which means that from trial to trial, the input into this neuron due to these learned connections is incredibly reliable. Just as the monkey's arm movement's incredibly reliable, it's going to be this periodic orbit in this constrained space that's not going to really wobble very much. But if we take a look at the trial-to-trial variability in the synaptic input due to the random connectivity matrix, this varied very, very strongly from trial to trial. And this is precisely what the model was designed to do. We included those random synaptic connections because we knew it would do this. And so the input due to this other matrix that is roughly orthogonal to the one that we constructed will give rise to these random fluctuations in its input. And the net effect of that is to give you something that looks like a neuron's response where you have a reasonably consistent kind of firing rate, spiking activity from trial to trial that's superimposed on top of this is some amount of spiking variability that distorts that reliability. And this is exactly what the firing rate is of a neuron. So if we take a look back at our definition of a firing rate based on the statistical model, we always define them usually as a summation of some factors that give rise to an expected firing rate. And we think about that expected firing rate as being distorted by a sort of random process that gives rise to spiking variability. And we can see that these correspond to these two different parts of the model. where we take our factors and pass it through some nonlinearity here, which will give you something roughly like the expected firing rate of this neuron. And then we pass this, we can think of this expected firing rate as being passed through a noisy process to generate some spikes. Those spikes are actually, in fact, due to a completely deterministic activity pattern in an orthogonal direction to the factors, which gives rise to variability. Are there any questions about that?
Sorry, I think this is just my slowness. So the weights that correspond to what you are calling the random inputs, those are also allowed to change during your target fitting?
No. So those are held fixed. Those are held fixed, and they are not functional. They were included with the prior knowledge that doing so would produce the exact effect that we wanted, which is variability in spike. And we wanted to illustrate that these highly precise factors could coexist with that variability via this. Oh, yeah.
So, thank you for the, I have another question following up on this spike, right? Because this is a rotationary task. So there is a certain like intrinsic frequency associated with the monkey's movement.
Yeah.
Suppose there is a gap between the two rotationary movements.
Yeah.
So the intrinsic frequency is broken at that point.
Yeah.
It's be able to reconstruct that as well. Because at this point, it's not like maybe you have other supporting material like showing that this is learning the neuron activity, not just learning the frequency.
Yes. So I'm going to show a task in a moment where it's not purely periodic, and that will, I think, support this. And I think it also, there's perhaps a second sort of sub-question there, which is, would the firing rate that you kind of learn as this projection of the factors be accurate if there was no input? And it would. It would look like if essentially there was no factor input, you would just have only the random connectivity input. You would get spiking variability that would look like noise, and this neuron would actually have no firing. The firing rate would be 0, despite the fact that you're getting spikes from moment to moment due to this extra input. So we touched on that a little bit in the paper, which is a kind of weird paradoxical effect, which is that sometimes a neuron would spike, and if you were an experimentalist, you'd be like, oh, this neuron is spiking, it has a firing rate, but actually if the factors are not changing, that neuron's rate is 0. Despite the fact that it's an empiricist, you might say it's firing. It has a firing rate. That's maybe a little bit too heady for this talk, but we did talk about that a little bit in the paper, which I think is a kind of curious aspect of our definition of a firing rate. Yeah.
So just to follow up on that, so there's no bias function that adjusts that, right? If the rate is not meeting the rates that activate the function, is there like a bias function that will basically perhaps increase the rate, firing rate in your model? Or
there
I
is
not see that anywhere.
there is a bias function that we constructed into the model that I didn't go into the equations for, and it takes the form as a setting of the reversal potential for the voltage. We just essentially set that so that the spiking activity of the neuron kind of exists within a space that it's necessary, yeah, or just kind of conducive to doing the task that we want it to do. I think if we didn't include that, you could still do all this, it'd just be harder. Yeah.
There was no fine tuning of the random particles. We constrain radius.
No, so that does need to be chosen reasonably, that needs to be chosen with some care. And the intuition there is that if the radius of that is too small compared to the effective radius that's learned by the learn part, then the variability will be swamped by the learn part. And vice versa. If it's too large compared to the effective radius of the learn part, then the learning won't work. And so it's essentially a balancing act. You want to get those to be roughly correct. And that will also, I think, depend probably on the ratio of the number of neurons you have to the number of factors and the number of neurons you have at all.
So the fit with the lowering of the factor. Look at the systems, more.
Not with like extreme care. We computed the Fano factor and we saw that it was roughly in the ballpark. But we didn't. The distribution was, yeah, but not individual spike trains. We looked at the just to, we didn't want to, you know, overdo it, and we just kind of said, well, this is in the region, the domain of reasonableness. With one other kind of key kind of focus of this was you know, I think a real sticking point, as Bernardo said, was that historically as a field, we haven't really had a strong command of constructing these models. We wanted this model to be flexible, to learn all kinds of things, and we wanted to illustrate that it could. So another task that we trained this model to do was the kind of now ubiquitous reaching task that has been trained so many times in the literature for firing rate models. So this is a kind of classic motor task for monkeys that are reaching. The idea is that on a screen, the monkey sees a bunch of targets. It has to reach its arm out to each one of those different targets to receive a reward. And this has been modeled numerous times with firing rate models. We wanted to illustrate with a reasonably sized network, a reasonably small sized network. This is 1,200 spiking neurons. You can construct a model that can do this task with all of the kind of bells and whistles and that we had insisted. So we're going to generate heterogeneous response profiles, sufficient spiking variability to look like real neural data. And I wouldn't be here if this didn't work. So this works quite well. So what I'm showing you here is the EMG activity of the animal recorded when they're actually making these reaches. We construct this model to produce those EMGs. So I'm showing you that for two different EMG target outputs for one trial across all the different reaches. This model can perform this task pretty well. And then all the kind of features that we are hoping this model would emerge out of this model do. So what I'm showing you here is a single neuron firing rate, a single neuron spiking activity across all the different reach directions for 10 or I can't remember, 10, 15 trials, something like that. And you can see that there's trial-to-trial spiking variability that emerges due to the random connectivity and a sort of heterogeneous response profile that looks basically like an actual motor cortical neuron. So this neuron has kind of varied preparatory and movement-related activity. Again, if you showed this, if I slipped this into one of Mark Churchin's papers, you might not even notice that was not a real neuron. And that was kind of, you know, back to the real focus of this work. It was we viewed this as a way in which modeling can support the theoretical neuroscience community broadly. People, you know, Mark built an entire lab centered around the idea that doing dimensionality reduction on motor cortical data and decoding EMG activity from this is a sensible thing to do. How do we know that? Well, we can build a model that kind of walks like a duck and quacks like a duck. So if it does all those things, then it works the same way. And so that's pretty much all in the past. Yeah. Yeah, so there was a two-dimensional input to essentially describe the coordinates on a two-dimensional space. I mean, there's, you know, there's a little bit of like baking this in to get the preparatory activity to look basically how it does. And, you know, we felt guiltless about that because part of what we wanted to express was that we understood sufficiently how to engineer the system to get it to look like data. Yeah.
The J factor is static as well. Only static external input.
Yes. So in the way that this model works for this task, this is very echoey. Down a little bit. There is during the preparatory period, there actually is the factor activity is 0. So the only inputs into the model at that moment are external inputs, not due to feedback that is learned. So this response here is actually due exclusively due to external input. And then the model has learned that after the certain amount of time passes, after that external input comes on, it should trigger a movement through the factor input.
Not explicit go.
There's no explicit go. Yeah.
But it can be something that this task does not need very high dimensional proof of outputs. So I was wondering why we did 1,200 neurons for this very actually large range in the number of neurons you need.
Yeah. So the main reason you need 1,200 neurons for these types of tasks is that the noise in the spiking is so large that you need a sufficient number of them to generate those factors that you need to. I don't know the exact scaling there. This is perhaps something that would be interesting to explore. Haim maybe wants to do it. But yeah, there's probably some kind of scaling there to describe exactly how many, what's the relationship between the number of factors you need and the number of neurons that you need to construct those number of factors. But it's essentially the noise is so large. By design, we constructed it to be that large.
So if we are just building the same as the rate-based model, like the abstract model in one.
Yeah, you could use it like 10 neurons or probably fewer than, yeah.
Many factors we never used.
This one is 37 for a reason that is very specific and that I won't belabor. We can talk offline about that if you want. Because I didn't exactly tell you how I got the factors from this one. And the short answer is I got them via a rate model, which is essentially works great. And we illustrate in the paper a few different ways you could generate them. For example, you could train a rate model with backpropagation, which we know how to do really well right now. That constructs factors really well. And essentially, I could take an MNIST model, trained it with backprop, and then insert it into the spiking model through this learning. So work that we've started to work on in my lab now is the idea that this is all very cute and all, but neurons, neural circuits do more than one thing. They usually do many things. So we wanted to start constructing models that do multiple tasks. So the idea is that there's some input that comes into this circuit and is telling the circuit it should do task 1 out of however many tasks that you want it to do. And we designed this model in a very specific way such that we wanted the factors that implement one task to live in a space orthogonal to the factors that live in a different task. Now, of course, I think it's a very interesting question of whether or not separate tasks might share some or many factors, but we didn't want to answer that question right here. We mostly wanted to see what are the consequences of doing more than one task, and how does that how can the models, these models and the notions of factors help us understand what's going on. And so we constructed a task we constructed a model that does both of these tasks, the reaching task that I just described to you, and the cycling task. And so I'm going to show you a little bit of what those data look like here. So this single model is being instructed at this moment to do the cycling task. And what I'm showing you on the right is what the factors look like when in the cycling subspace and in the reaching subspace when the animal should be cycling. And so not surprising, when the animal should be cycling, the factors in the cycling subspace are large and robust, and the factors in the reaching subspace are tiny. So because the animal shouldn't be reaching now, it's not supposed to be. And then vice versa, when the animal is performing the reaching task, the factors in the reaching subspace are large and implementing the computation. Factors in the cycling subspace are small and not doing anything. And what we're starting to think about is perhaps the idea that spiking noise might be, so let me just back up for 1 second. The way that we introduced spiking noise in the model previously was by including a random synaptic connectivity, which worked, but you might say to yourself, is that how and why you think neurons spike variably? So the question we'd like to consider here is perhaps spiking noise is due to the fact that when you construct a model that does more than one thing, and you start to pack more and more things into a model that is capacity limited, the consequence of that is that you're going to get spiking variability. So the idea here is, imagine I have this state space over here, and there's essentially two orthogonal spaces. There's the cycling subspace and the reaching subspace. So when I'm doing the cycling task, all of my activity should be living in the cycling space. It shouldn't deviate out of that space. And anytime it does deviate out of that space, it's going to start introducing spiking variability that is going to be to the detriment of cycling. And so you can think about the activity when I'm correctly doing my cycling, I'm going to be moving through this cycling subspace, and that's going to be the correct thing to do. And then occasionally, I'm going to deviate outside of that into the reaching subspace, and that's going to start to produce spiking variability during my cycling task, and vice versa for the reaching task. And so obviously, when you're doing two tasks, that's not going to be perhaps super detrimental, but of course, my motor system can do many, many things. And so perhaps the price that we pay for constructing a multi-task system is that we start to reduce the fidelity with which we can do any individual task. And this should have roughly the flavor of a Hopfield network. We know that if you pack more and more memories into a Hopfield network and we begin to breach the capacity of the Hopfield network, the fidelity of memorizing any individual memory will start to degrade. So we're constructing these models now. It's not clear to me that we can do any robust theory in this space, But I do think it would be introduce a new interesting hypothesis as to what is the actual source of spiking variability in motor cortex. Yes.
Clarification for this construction. So when you do multitask, you share the latent factors between the task, and there's capacity limit on how many factors include.
Yes.
Is there, do you observe the size of the limitation based on the capacity change?
No, we haven't. So the way we built this model was explicitly, we built it explicitly so that the factors were orthogonal to each other for these two different tasks. I think that you could reasonably build a model that had shared factors, and that seems like a sensible thing to do. Like if I'm moving my arms or moving my legs, like there's probably some motor primitive that's being shared across these multiple different tasks. But at some point, like they have to differentiate themselves enough. They are going to require other signals to differentiate them. And as this system begins to do more and more things, eventually it's going to hit up on a capacity limit where signals are going to inadvertently leak through these other factors for doing other tasks. And that's going to look like variability for the task that you're actually executing.
It's actually made 2J factors. Matrices, one for cycling and one for reaching. Switch between modes by setting putting one in and the other one in, or is there something else?
There's an external signal that kind of tells it. It's like if you were a monkey and I asked you to do cycling now and then do something like a context view. They do this one or do this one. It's going to switch you between these two different states. And I mean, really, I think about this more as like not 2J facts, but like 2-factor matrices, but 2 matrices that are just stacked on each other. And essentially, I think, the idea, we haven't actually constructed it yet, but as that matrix grows larger and larger and all those are orthogonal, that's going to roughly start to look like a random. So there would be no JFAC anymore. That would just be a random matrix. And the directions that are in that random matrix for tasks that you're not doing would do the exact same thing that the random was doing in the model that we built. Yeah.
Did you generate those latent factors again through like a rate network and then plug them back in, or was it like?
So the cycling factors came from the data as before, and the reaching factors came from the rate model like I did. I really just took the two models and I mashed them together. Yeah. Yeah.
The random connectivity model, I'm curious, can you resample the random connectivity and get the same output figure as well?
Without retraining the model? It's sensitive to it for sure. I don't know exactly how sensitive to it. I think it won't be catastrophic, but it will be sensitive. I mean, because it's trained in the face of that specific sample that you used, and it's certainly, I think, rotating itself away from really, really deleterious direction. And so if you resampled it, the possibility of you aligning that deleterious direction with the factor directions would be constant. Are there any other questions? I'm going to move on just to a few other things. So you said we have like a real hour and a half. Like, is that for realsies or? I'm going to move on just to some new things that we're doing in the lab and a little bit of work that I did on the postdoc. If anyone wants to keep, this is essentially the end of this part of the talk about spiking neural networks. If there's other questions, I'm happy to take it. Otherwise, I'll just move on to some new stuff. Yeah.
So in terms of number of neurons, I think you mentioned that if we had great neurons for it to replicate, I don't know, 12 latent factors, we would have needed I don't know, on the order of 10 neurons. So from your work or maybe the ongoing multitasking work, is there a sense where having spiking neurons will offer an advantage? I mean, because it sounded like for the 12 latent factor thing, having spiking neurons just meant that we need a lot more neurons.
I think in the paradigm that we've designed, spikes only make things work. We have essentially adopted a rate coding paradigm, but we wanted to construct rates out of actual spiking. And so I think for other systems where they maybe use a code that is more specific to spike timing, this framework will change a little bit, and that will be, I think, a slightly different situation. But for this general paradigm, we were really coming into it with the perspective that this system is performing rate coding, but we wanted to understand how it could actually do that with a reasonably sized spiking model. So previous work has, just to give a little bit of history and context, the evolution of trying to actually construct these models, spiking models, like previous incarnations of this would just take individual rate models. Like say you had a 100 neuron rate model, and they would take each individual neuron and replace it by an entire population of spiking neurons, which seemed like a sensible thing to do. But that clearly grows very quickly. And the insight that we had is that if you think about it in the factor space instead, we know that each one of those rate neurons is not entirely uncorrelated from each other. So if you just identify the relative kind of space of dynamics in the factor space and align that with the relative space of factors in the spiking space, you can do this much more. Okay. So I'm happy to move on to some other stuff that we're working on in the lab now. I think it's also very exciting, a slightly different flavor of things, not quite the same as the mechanistic models. But just to summarize, we developed a pretty flexible method for constructing functional spiking models. We built them so that the latent factors are an abstraction, a powerful abstraction that's biophysically real in the neural circuit. It's not just pretty pictures that we draw. This is essentially a signal that's embedded within the synaptic connectivity of a neural circuit. And that takes the form of a low-rank synaptic connection that allows this system to produce reliable spiking activity, which we typically call a firing rate. So this was the bulk of this work was done during my PhD and then into my postdoc, and it really kind of came about because my PhD advisor Larry Abbott and I were having like a little bit of a crisis of thinking about, well, these low dimensional representations of data are seeming like really useful, but does this have any real relevance to how a neural circuit works? And now that I've convinced myself that this seems to be a real bona fide abstraction that a neural circuit can implement, I felt free to move on into the machine learning space of applying these types of latent variable models to data to try to understand how neural circuits work. So what I did during my postdoc and what a lot of the work I'm continuing on in my lab at BU is focusing on machine learning methods for analyzing neural data so that we can construct these types of abstract models and understand how neural circuits are performing the computations that we're doing. So I'll tell you a little bit about the two different projects that are going on in my lab now in decision making, and a little bit of new work that we're starting to do in the olfactory system in collaboration with investigators who are studying mosquitoes. So I'll start with a little bit of an overview of some modeling that we've been doing in the decision-making system. And decision-making seemed like a great place for machine learning methods for understanding neural population activity, because so much of the signals that are relevant to decision-making are not externally observable. They're happening within the brain of an animal, so we're going to have to infer them through a cleverly designed machine learning model. So this is the kind of task that I'm thinking about these models in. This is an evidence accumulation task. This work was done in the lab of Carlos Brody at Princeton. And so many of you are probably familiar with the evidence accumulation paradigm from the Brody lab and others. But the idea is that an animal will poke its nose into a central port, and they'll hear a series of auditory clicks on the left and the right. And what the animal has to do is essentially count how many clicks were on the left, how many clicks are on the right, and make a choice to one port or the other, depending on if there was a greater number of clicks on the left or the right. And then my colleagues in the Brody lab go in and record from a variety of different brain regions to understand how this animal performs this task and how these brain regions are going to interact with each other. So the Brody lab has been doing a lot of different recordings in many different brain regions. I'm going to focus on 2 brain regions for this talk because we found some pretty interesting signals in these different brain regions that were interesting and relevant to how the animal is making decisions. These 2 regions are the frontal orienting fields, which is roughly the rodent homologue of the frontal eye fields in the primate, and then the striatum, which is the input into the basal ganglia. So I'm going to move pretty briskly through the machine learning stuff. I'm happy I have some extra slides if anyone's curious about the method, but I was not exactly clear how much time I would have. So we developed a machine learning paradigm for essentially implementing the drift diffusion model, which is sort of a classic model for how animals might accumulate evidence in order to make a decision. So we essentially took a classic model and sort of machine learningified it so we can perform statistical inference and learn that model directly from data. And there's a few kind of key components of this model. It's a latent variable model, which is sort of the statistical language for manifold or low dimensional dynamics. And so this model takes the form of a temporal accumulator, which I'm showing you here in these colored lines. And that will integrate information from the sensory periphery and tell you at a moment-by-moment basis which one of the two choices the animal is likely to make. This is a parameterized model, which allows us to learn a few important parameters from the model about what is going on in this animal's head when it's experiencing these sensory stimuli. And the way that we're going to fit this model is by making a bunch of different types of observations that will allow us to perform inference and learn what the latent variable model, this latent variable model is. So we're going to use neural spike trains recorded from these two different brain regions and the behavioral decision that the animal made at the end of the trial. Did I go left or did I go right? This should report to us something about what was going on in this animal's brain when it was making this decision, and we'll do normal statistical inference using essentially gradient descent on this model to identify the parameters of the model. So as I said, there's a little bit of interesting machine learning going on here. We essentially like converted a drift diffusion model into a discrete approximation to a Fokker-Planck equation, which will essentially take the form of a hidden Markov model, which is a machine learning model where you have relatively sophisticated handle on optimizing. And if anyone is curious about those points, I'm happy to talk about that offline. But so when we fit this model, what does this allow us to do? What kind of scientific questions can we answer? So there's really kind of two key parameters that jumped out of this model when we fit it. One is the integration timescale. This tells us how forgetful each brain region is in terms of the activity that its neurons are reflecting. And the other is the commitment boundary. This is a parameter in the model that tells us at what point the animal has made a commitment to make a choice, no matter what information comes into the animal's sensory system after that commitment has been made. And what we found was that the two different brain regions that we were studying did quite different types of forms of evidence accumulation that we could learn directly from fitting this model. So the FOF seemed to express an extremely low commitment boundary. And what I'm showing you on the left here are samples from the model once it's been fit to data, showing you the drift diffusion process that would be generated from a model fit to the maximum likelihood parameters from that neuron, that animal. And so what you can see is that the FOF seems to express an accumulator where the animal will very prematurely commit to whatever decision it's going to make, even before the sensory stimulus has finished. Where the striatum seems to show quite a different type of accumulation process, which is a very high boundary, almost as high as it could be, which essentially means that the neurons in the striatum continue to listen to sensory information for as long as they possibly can before they commit to a choice. The same basic strategy seemed to play out in the integration timescale, which is essentially how forgetful each brain region is. What we found in the FOF was that the integration timescale was positive, which essentially means that the animal is not forgetting things, but essentially amplifying early information at the beginning of the trial, which is essentially an impulsive strategy. If I heard one piece of sensory information very early on, that piece of sensory information will amplify as time goes on and make me more likely to make a choice to the direction that sort of felt very, very early on. And the striatum seemed to do the exact opposite. It was a leaky integrator, just barely, which meant that it was slowly forgetting information as time went on, but never really prematurely committing to a choice. So we found this to be particularly surprising. It means the animal somehow combines these two pieces of information from these two different brain regions to make its choice. And we don't quite yet understand exactly how that's working. We're thinking about building multi-region models of this type to understand how these two different brain regions interact. They do support some curious inactivation studies that the Brody Lab has undertaken. For example, it's known that if you inactivate the FOF, the animals generally become leakier in how they remember information from a behavioral perspective. So if you look at how they're making choices without any information about their neural activity, they seem to be more forgetful. And that would perhaps suggest to you that if I remove the FOF, that's causing the animal to be more forgetful. But what it seems to suggest here is that the animal is not getting more forgetful, it's actually becoming less impulsive, because I've kind of pulled out this impulsive part of its brain, and therefore it's kind of relaxed more, which is kind of a perhaps counterintuitive intuition as to what's going on in the FOF, but that's what the data seems to suggest.
Make sure I understand. So upper left, red curves, that's a purely searching perspective. The animal doesn't necessarily follow behavioral level, it goes down.
They do not, yeah. So at a behavioral level, they're actually almost perfect. They're very tuned into what the stimulus is, and they make a choice almost using every piece of sensory information that they can. But the FOF doesn't seem to be. It seems to be like wanting to make a choice, and someone is stopping him. Someone is stopping that part of its brain from making.
So the way those two look, you actually cannot predict the animal's behavior from inspiration found in either of those structures, or you can use.
No, yeah, and even using them together, it doesn't quite work just right. So we know there's other brain regions involved. We've been fitting other types of data to it too, but if I just looked at the FOF, I would be wrong. It would tell me the wrong answer about what the animal is doing. Yeah.
I'm really curious about the FOF, the two plots, basically, they're qualitatively saying the same thing. I think that sort of amplify the earlier ones. It's kind of qualitatively similar to earlier times of continuous state. So are those like, are you sure about like the parameters that you fit? They could be like degeneracy.
There is a, yes. There is a little bit of degeneracy. So when we fit this, we usually will compute error bars on the parameter estimation by looking at the curvature of the log likelihood. And we do see a little bit of trade-off between these parameters. I can't remember exactly what it is for those, but we investigated it. And so there is a little bit of play. I mean, the parameters I'm showing you here exactly, but actually these, so I am showing the error bars here. They're just so tiny you can't see them. So there is a small error bar here on the parameter estimate for that. And there's a small error bar on the parameter estimate for that. So at least for this data, they're pretty accurate. But in general, we looked at the curvature to ensure that be certain we how accurately we were estimating. One branch of work that my lab at BU has started to undertake is following up on, I think, one of the most interesting aspects, one of the kind of most interesting threads of research going on in decision making right now, which is a pretty clear, I think, evidence that animals are changing decision making strategy on a moment by moment basis. So what I'm showing you here is results from Jonathan Pillow's group. This is this Ashwood paper from 2022. And what they found is that just observing what animals are doing on a purely behavioral level, they were able to fit switching type models and illustrate that these animals are switching between different types of decision modes in a moment by moment basis. The animals essentially periodically in an engaged mode where it's performing the task, and then will occasionally go into some kind of lapse mode where it's kind of checked out and not really doing anything related to the task anymore. And they show that the animals at a behavioral level seem to clearly be doing this. They go through long periods of being engaged and then being disengaged. And the natural question I had is, well, what strategy parameters that we know from a DDM are changing at these moments in time? So that led us to develop what we're working on presently, which is what we call a HMM DDM. So it's a drift diffusion model with an HMM underneath it, which allows you to switch between different drift diffusion models as a function of state. And the basic idea here is that perhaps when I'm in one trial, I'm using one drift diffusion model. On a second trial, maybe for whatever reason, I didn't get the reward that I was expecting. My boundary goes down, and now I'm going to become even more impulsive than I just was a moment ago. And we've started fitting this model. This is work that's being done by my graduate student, Ryan, in collaboration with Ben Scott, who's a faculty member at BU. And as I said, we're essentially putting an HMM directly under our drift diffusion model. And we started fitting this data to a variety of different brain regions, and we find that it's very, very clearly does a better job at describing what's going on when animals are making decisions. So what I'm showing you here is the posterior state estimate for which state the animal is in as a function of trial number. And you can see that the animals very quickly, and what that means is that if this red curve is down near 0, it's very unlikely that I'm in one of these two states. And if it's up near blue, I'm definitely in that state. And you can see on a trial by trial basis, the animal is pretty rapidly moving between these two different drift diffusion states. If I take a state-weighted psychometric function, if I compute a state-weighted psychometric curve, you can see that the animal is in state 2, clearly performing the task much worse than when it's in state 1. And if I do a state-dependent, if I compute a state-dependent PSTH for all the neurons that I use to fit this model, you can see that clearly this neuron is firing rate depends on which state it's in. So this neuron's firing rate will and so what I'm showing you here is the firing rate is a function of time for conditions when there's strong rightward evidence, strong leftward evidence, and weak rightward evidence, and weak leftward evidence. And you can see when the animal is in state one, performing the task very well, this neuron is much more responsive to strong rightward evidence than strong. This neuron is much more responsive to strong rightward evidence than when it's in state two. So this is all kind of checks out, and we're continuing to develop this model to understand how we might be able to use it to more directly understand state-dependent decision-making as animals are performing these types of decision-making tasks. We are about to open source some Julia code. If there's any Julia aficionados out there, we are pretty big into Julia in the DePasquale lab. We're about to open source a code for fitting all different types of state space models in Julia. So if you're curious about that, please reach out to me. Happy to share it. So with that, I'd just like to move on to some kind of work that my lab is undertaking. It's pretty different than what I've done in the past, but I'm pretty interested in it, and I'm happy to hear what folks think about it here. And so when I started my lab at BU, I kind of seemed like a moment, take a pause and say, like, what's going on in the world? What do I want to start studying? What's the landscape of neuroscience look like? And I took a look and I said, what's the what is our success story? Like, what's the best job that we've done so far? And I think that it's pretty clear that the best job that we've done in computational neuroscience at the present is in vision. And the reason I think that is because we can essentially construct purely artificial models that are roughly biological and give them a task that we know part of the brain really does, which is, like, for example, take this image and classify it into a category. And we can get these models to do these tasks almost as well as we can get people to do them. And what's even more interesting about this is that if we examine these models layer by layer, they seem to roughly look like the processing of visual information that we experience layer by layer. What I'm showing you over here is the activity of a real and a simulated neuron in this model. And it's showing you the firing rate of that neuron as a function of the stimulus that's being shown. And this particular model was not trained to make sure that the neurons match, the simulated neurons match the real neurons neuron for neuron. This model was simply trained to perform object recognition. And just by pure chance, what pops out in something like IT in the model looks like an IT neuron. And to me, I think this, in my opinion, takes the cake. I think this is the best job we've done. So pretty cool. And I started to think to myself, well, you know what, this kind of reminds me of something. Like, what's something that just like is always just winning, constantly crushing it all the time? And it really just reminded me back to thinking about New York of the Yankees. And I said, you know what, I'm going to Boston. I can't just stay in the space of winning. Where, you know, what's that group that really needs a little bit of extra help? I realized that this joke is kind of not working anymore, mostly because the Red Sox are kind of good now, you know. But like, as I said, I'm kind of old school. And so in my mind, you know, 2004 never happened. Or was it 2007, whenever they won? 2004? I don't know. Another way I started to look at this is I started to say, well, let's take a look at vision models as a function of time and see how well they are doing against a human level performance. And it's a little bit tricky to establish exactly what human level performance is for classification on ImageNet, but my understanding is that it's roughly around 5% error for a human. And so that means about now, OmniVect is about at basically a human performance level. So just again, I think we've done a pretty good job in vision. So this led me to sort of think about where are we not doing a great job? Where is places where the models that we build are just really not even close to the kinds of models, the kind of biological models that we already have. And I think the space that we can really do a much better job is in olfaction. I think this is an extremely interesting sensory system that essentially, if you gave humans the option to classify different smells into different different classes, we are just not even close to building a robot that can do it as well as this dog. So clearly, we have quite a bit of work to do here, and I think there's a lot of work that we can take a lot of a lot of what the progress that we've seen in vision and try to understand how we might move it over to another sensory modality like olfactory. So this is work that I'm doing in collaboration with Meg Younger, who's a mosquito researcher at BU, and this is my first graduate student, Grant, at BU. And we are starting to work in the space of graph neural networks, which is, I think, a really exciting space in the machine learning world. So, and I think it's particularly exciting because the structure of graph neural networks just really lends themselves very well to the space of computational chemistry, where a huge amount of data is now being generated and can be, I think, applied. These types of models can be applied really, really successfully to try to understand how chemicals interact with all forms of biology, including our olfactory system. So for those of you who don't know, graph neural network is basically very similar to a convolutional neural network, where you, for example, for a chemical, you would convert each atom into a node, and each edge, each bond between atoms into an edge, and you could imbue each one of those nodes with a feature vector of information about what that atom is. And for example, I could say, well, for this I can one-hot encode what this atom is, what its hybridization is, what its valence is, it has an aromatic ring, all these different important properties about what this chemical structure is. And then you can pass it through essentially a deep neural network, much like a convolutional neural network. And this will take those features that you've given the graph and process them into a different set of features that will hopefully be helpful for doing something else. And so the task that we would like to try to do is, can we take the chemical features of a variety of different odorants and predict how olfactory sensory neurons respond to those odorants? So essentially, we want to see if we can play out the success of the visual deep learning world in the olfactory system. And one problem that is sort of immediately apparent is that for this kind of task is still pretty scarce. For example, like we don't have nearly as much data compared to the visual system. So how can we kind of circumvent that problem? So again, taking a page out of the deep learning playbook for vision, we know that self-supervised learning is a really powerful method for constructing neural network models when you don't have a lot of data. And so the basic idea behind self-supervised learning is you take a data set that doesn't have labels, and you create a bunch of synthetic labels that allows you to learn the structure of your data in a way that will hopefully be better for doing something else that you want to do. So for example, what we would do is take a large data set of chemicals, which is freely, abundantly available. I don't need to know if olfactory neurons respond to those chemicals. I just need to know what their chemical structure is. And then I can derive a bunch of tasks, like, for example, removing an atom from this molecule and see if I can predict that atom based on the adjacent atoms, or remove a bond from an atom and see if I can predict it from the adjacent bonds. And I have 15 million chemicals in a database. I can pre-train a model and see how well that allows me to learn the overall chemical structure of odorous molecules. And then from there, I can now take that trained model and take my much smaller data set and see if it allows me to improve my ability to predict on this very, very tiny data set. So we took an existing data set called the DOOR data set. It's a reasonably tiny data set of what odorants bind to which olfactory receptors in the fruit fly. It's about 5,000 data points, which is pretty tiny compared to the kind of data sets we might need. And what we found is that this actually worked pretty well. So we trained our model using pre-training. We saw a small improvement, a reasonably small improvement over the ability to predict olfactory sensory neuron responses than if we did no pre-training at all. So this is still early days for us, but I think it just, it illustrates that playing out much of the success we've seen in vision will allow us hopefully to make a little bit more progress on a problem that is, I think, in desperate need of some of these tools. So this is, again, I think this is a real kind of interesting movement away for me from spiking neural networks, but I think there's a lot of exciting machine learning work to do here. So as I said, my colleague Meg Younger is a mosquito researcher. Mosquitoes are an emerging model system in neuroscience that we don't really know much about. And she's especially interested in trying to understand which olfactory cues bind to which olfactory receptors in the mosquito And I think it should be pretty obvious for most of us who've ever been around a mosquito why you might want to understand what chemicals bind to them, because that's how they figure out where you are so they can bite you. And if you live, maybe not in this country, but some other country where disease is much more prevalent, you maybe recognize that mosquitoes kill more people on this planet than any other organism alive. So if we could begin to understand the olfactory system of the mosquito, we might be able to mitigate its global health effects on humans. And so we're beginning a project now where we're going to try to take these types of models that I just described and use them to initiate a large chemical screen of which odorants bind to which olfactory receptors in the mosquito. So this will take even a greater digression from the kind of work I've done in the past, where we're essentially going to train two models simultaneously. One is a graph neural network model that takes information about the chemical structure of odors. And the second is a protein language model, which is a large language model adapted for amino acid sequences for proteins, and try to predict which chemicals bind to which receptors on a large variety of existing olfactory data sets. And this will allow us to predict which chemicals are the best chemicals to start screening for mosquito olfactory receptors, to rapidly move through the space of chemicals and identify which receptors bind to which chemicals bind to which receptors. And so we are planning to kind of build a machine learning dependent chemical screen to try to, as rapidly as possible, identify all of the olfactory receptors that bind, all the olfactants, all the olfactory cues that bind to all the olfactory receptors in the mosquito. So with that, I'm happy to take more questions about any of the content. And as I said, it was really very, very exciting to come and give you guys talk about this work. This is my first talk as a new faculty member, so it will definitely go down in my mind for all time. These are my collaborators for the spiking neural network stuff, my PhD advisor, Larry, Mark Churchland, and this work was done with David Cicillo, who I think many of us know, who's at Stanford and also at Meta. My postdoctoral advisors, Jonathan Pillow and Carlos Brody, were working with me on the decision-making stuff. I had lots of collaborators over the years through all these projects, including Kanika, who's here but not here. And I have a new lab going now at BU. I'm growing quickly. I got 6 students, lots of collaborations with a variety of different investigators at BU and other locations. And as I said, it's fun times there. This is our very, very first lab meeting, a picture of our very first lab meeting, which was kind of special. So thanks, and I'm happy to take more questions.
And of course, there are sources of noise that practically probability is on one. So how do the models in the first have to cost grade or deal with noise or? Yeah, we never really explored that. I think it's an important point that we should definitely follow up on. And the only reason that we really didn't is because we knew it would be a problem, and we really wanted to kind of focus the attention on how the random connectivity would give rise to something that looks seemingly stochastic. But I think probably many of those questions, I think there's probably 2 possibilities. One is that like the random connectivity, the structured deterministic noise and the stochastic noise interact with each other in a really bad way that makes things really bad. Or they don't really interact at all, and you could probably just calculate some kind of like, how bad the noise might be as you increase its magnitude in PLM, where the noise is basically. I mean, a long time ago, actually, my very first project during my PhD was constructing rate models, where we added on top of them Poisson-like noise. So essentially like melding a firing rate model with like a GLM. And we saw that noise can actually be like pretty bad. So it's possible that it's, it has a strong impact, especially might be amplified by the chaos. But we haven't really. It's interesting being in a biomedical engineering department, like all the students want to build machine learning models now. They all want to build deep neural networks. They all want to like do synthetic biology. And then I'm constantly like, hey, let's build these like, spiking neural networks. And people are like, no, build large language models. I'm like, okay, sounds good. Yeah.
So you talked about the greatest success being vision, and the reason being that all this stuff is like simulation. So is your goal with the olfactory work to build models to simulate real neurons, or do you think there's something about the graph neural networks will allow you to like parse, like understand grammar.
I think it's a little bit of both. I think ideally we would like to build a like 3-layer or 4-layer neural network of the early olfactory system of a few different types of organisms. I think that it's possible that the graph neural network representation might give us greater insight into how to think about tuning in those early layers. So I think it could be a foothold into doing that, but it's not exactly clear to me how to do that quite yet. I do think that the graph neural network work right now is just really helpful for trying to understand what is the the latent representation of odor at various stages in the olfactory system. And there's some interesting work. So I just sort of began a collaboration with Betty Hong at Caltech, and she's generating really interesting results from Drosophila, looking at how representation, essentially like representational geometry of olfactory of smell changes as you move deeper and deeper into the neural circuit of the olfactory system. And I think graph neural networks will give us a little bit of insight into what's going on. So, you know, a few different people now have found that as you get to essentially like the mushroom body, which is like the association cortex for Drosophila, the graph representation, which really represents chemicals, is kind of gone. So it's really by 3 synapses in that these animals don't seem to really even care about chemicals anymore. They seem to be much more interested into what those smells might tell me about what's going on with the food that I might go eat. So I think there's a lot of interesting work.
Good question. In the first part of your talk, you put forward this hypothesis that the variability emerges because the system is trying to do multiple tasks.
Mm-hmm.
But at the level of single neurons, by physical level, you have many elements which cause the noise to emerge for each signal. For example, you may have a stochastic resonance and ion channels.
Yeah.
So somehow even there, actually, you have a lot of noise.
Yeah.
Right.
Yeah.
So and it may be, but actually the studies that show stochastic resonance is more prominent in sensory neurons, and for very good reason, because you want to amplify the system to the noise, the SNR. You want to amplify it towards the end of a large network, let's say in human brain, that's the cortex. You may not want as much noise, so there must be a difference in terms of at what level of the system you're looking at. That variability at the level of biophysical neurons to the network level could be very different.
Yeah, I think that's totally right. I think, for me, I'm curious about this because to my knowledge, I don't think this has been posited as a mechanism for variability. I mean, as I said, it's sort of very similar to the notion of memory degradation in hot field networks, but I don't know that it's been introduced in this idea of spiking models and this idea of packing in multiple kind of like manifolds for representing different tasks. But I think, I feel like the one thing that I think that is becoming more clear to me all the time is that like every individual part of the brain, every individual organism's brain is solving so many different tasks in so many unique ways, and looking at how those things are, the differences that are occurring in different brain regions is super important. And like, you know, those kinds of effects. Where is noise coming from? Where's the source of noise, and what does that tell us about what that's doing?
Second question. For your graph neural network, when you're basically building it, you said based on like 5,000 molecules or something.
Yeah.
Okay, so do you have any way to check and see these molecules physically like possible to make them? Do they even exist in nature?
Yeah.
Can you build it?
That's a really great question.
Try to see, okay, what sort of like labeling you can add to them, et cetera, et cetera.
So that's a really great question. So all of the molecules we use here are naturally occurring. So they come from 2 data sets. So the first data set was a large data set of small molecules, and those are all existing. The second one is from molecules that have been shown to Drosophila, so we know that those both exist. But I do think that the question of synthesizing, again, looking at vision, Jim DeCarlo's work has shown us that if you build a sufficiently accurate surrogate model of visual system, you might be able to use that to synthesize artificial stimuli that maximally cause real neurons to respond. And I think if you play that out in the olfactory system, I think you might find yourself in a very, very weird space where you're starting to synthesize novel compounds that are causing neurons to become maximally active. And I think in the context of mosquito research, that's very interesting because that is essentially what I think many people would like to design, which is like get these mosquitoes the hell away from them. So it's, fascinating being, and as I said, the bulk of the researchers in biomedical engineering at BU are in synthetic biology. So I started to chat with them about this because I think it's really too. I mean, from the neuroscience perspective, it's curious to think about how can I cause olfactory neurons to become maximally responsive. But if I come up with a novel compound that doesn't exist that causes these olfactory neurons to become super responsive and gets mosquitoes to fly away, they're like, I'm going to probably go build that. Sell it.
Okay, stop there.
Yeah.
Thank you.